Okay, so we've worked our way through a lot of bioenergetics up to this point. But what I want to do is I want to bring it all back together again, like one big happy family. So hopefully you're not too intimidated when you see this chart anymore. You probably have a little bit of worry that I'm going to ask you every little piece of this, but I'm not, because you're not biochemistry majors. We need to know the general overview of this in order for us to adapt it to exercise. So we're going to look at this in a bunch of chunks. The first thing I want to do is I want to get rid of this table here. So let's look at that. Here's what we have in that table. We have two distinct categories of substances that are used during metabolism. In the green, you'll notice, are all enzymes. How do we know they're enzymes? Because they all end in ase. We have PFK, which is phosphofructokinase. We have phosphorylase. We have HK, which is hexokinase. We also see one more of these enzymes present down here in lipase. So these are all enzymes. They're breaking something down. Let's go back to this chart, and in blue, you're going to see all of the hormones that are present during metabolism. We have epinephrine, fight or flight response. We have insulin, which helps recover glucose from the bloodstream, glucagon, and growth hormones. We're not going to hit on glucagon and growth hormones at this point. What's really important for you is to identify epinephrine and insulin. We can see those popping around here, but we'll get back to those later in this video. So let's get rid of that chart. Cleans it up a little bit. So now we're just looking at the overall process of metabolism, including glycolysis, oxidation, and beta oxidation. But what I want to do is I want to break this diagram down into 5 or 6 distinct chunks. The first thing we're going to look at are the major structures involved in metabolism. So if we cut away all the fat, we see that there really are 4 main things we're looking at in terms of structures. We have adipocytes or fat cells. We have the blood vessels that circulate blood around the body. They come in contact with cells, and we're going to primarily deal with the cell cytoplasm. And within that cytoplasm, we have mitochondria. These mitochondria proliferate throughout the cell. We have more than one in each of our cells, and there are varying qualities. The next thing we want to break down from that large diagram are where do we get the energy from? So let's add in what are the things we break down in order to get energy? Well, we have fats, lipids, and then of course we have glucose and we have glycogen. Remember, glycogen is simply the storaged form of glucose. Glucose is available freely in the blood, while glycogen is located throughout the body. Primarily we see that in the muscle cells themselves, and then we see it also carried away in the liver. Let's isolate those hormones and enzymes that we talked about previously. Let's see where they start to come into play. Now this is important because a hormone like epinephrine, our fight or flight response, has 2 distinct actions. We see it acting in the blood here in order to harvest fat. We also see it activating here to help bring, you remember glycogen is here, to help bring glycogen down. Insulin, located in the blood, helps bring that glucose into the cell. In green, we can see all of our enzymes and where they act. Now the key thing about this slide is anything in blue or green on this slide acts as a switch, meaning we need its presence in order to drive metabolism. And if we don't have one of these, we're not going to drive metabolism. For example, if we lack insulin, like in diabetes, we can't bring that glucose into the cell, and so we never even get glycolysis started. Also, if we have a problem with phosphorylase or synthetase, we can't actively store or break down glycogen. That gives us a problem because then all we have is energy coming from this side. Alternatively, if we lack hexokinase or phosphofructokinase, we can't drive through glycolysis here. Okay, so these are just different conditions that you have to be aware of. The primary one that you'll encounter is people who lack insulin. The next thing we want to identify on that chart is where all the energy is. Pure energy, ATP. This is the goal. What we have to remember though is that we have an investment of ATP in order to kickstart this whole engine. So we lose 1 ATP here, we lose 1 ATP here. Only then do we start to recover ATP throughout the bottom half of this diagram. 15, 3, 1, and 1. We're going to get into that in a little more detail in just a second. Now, we've been through several items in these slides so far. What we want to do is we want to make sure that you understand that we have structures, we have sources of energy, we have hormones and enzymes, we have energy. But if you remember, that diagram was much more complex. So we have everything else that happens. These are considered all intermediaries or other substances that contribute. I'll talk about a couple of these, but not too many in detail. What we can see here is calcium. Calcium is present and able to stimulate that drive from glycogen back down into the beginning of glycolysis. So if we look very quickly here, here's our calcium, and this helps drive the harvesting of glycogen. Epinephrine also helps drive that harvesting of glycogen. This is important because calcium is released when muscles contract, epinephrine is released in fight or flight. So once you see something chasing you, you get scared, your muscles start contracting, you can start pulling all that glycogen for that excess energy. Okay? So there's a lot of other substances that start to happen. Pyruvic acid, lactic acid, acetyl, everything that dumps into the cycles. We can see that we produce adenosine diphosphate when we harvest that ATP. Okay, so really we've got everything else but the kitchen sink, but we might as well, you know, we might as well just throw it in there. Okay, so now we've got the kitchen sink too. All right, so one other thing that you need to keep in mind is right here in this square. Okay, what I've noted here in this diagram is a triangle, and this triangle simply indicates that from this one glucose molecule, if we follow that arrow through, it becomes glucose 6-phosphate. Phosphofructokinase has an action on it. We lose an ATP. It feeds into this triangle, and it's going to feed into what we know as two identical branches. So if we look at the full diagram from this, here's that same triangle with two legs coming down on either side. So actually, where we see 3, 4, 5 ATP on this initial diagram, we actually get 5 ATP harvested on both sides of this process. So that gives us 10 total ATP, again, 5 from each side. Don't forget that we have two sides driving this. For the simplicity's sake of this graph, I've only included one of those legs processing down, but there are in fact two of them. Keep that in mind. So the next thing we want to talk about is actually the process of glycolysis. So let's pull away a lot of the other stuff that's happening here, and let's talk simply about glycolysis, the process of taking either one molecule of glucose that's in the blood or one stored form of glucose in glycogen. In the presence of insulin, glucose goes into the cell, hexokinase works on it, takes an ATP to pay for it, becomes glucose 6-phosphate. That glucose 6-phosphate gets worked on again, loses another ATP. So right here we can see that we're down 2 ATP under one molecule of glucose. From there, it goes down this triangle. There are two branches, and we just previously saw that in each of the branches we produce 10 ATP. That's why when we get to the end of glycolysis, from one molecule of glucose, we have 8 total ATP. The other way we can get there is by harvesting one glycogen molecule. Calcium from muscle contraction, epinephrine from fight or flight activates phosphorylase B, turns into phosphorylase A. From here, we start bringing glycogen down into glucose 1-phosphate. Phosphofructokinase acts on it, and we lose only one ATP. So right here is the difference. If we're coming in from glucose, we lose an early ATP here and a second down this branch. If we're coming in from glycogen, we don't lose that initial ATP, we only lose one. So you can see here from glycogen, it's minus 1 ATP. Even with glycogen though, we travel down into that triangle where there are two equal branches. We again produce 10 ATP. Therefore, our total gain is 9 ATP at the end of glycolysis of one glycogen molecule. It is only 8 ATP from glycolysis of one glucose molecule. This sums up glycolysis. At the end of glycolysis, we're left with either a pyruvic acid or a lactic acid. Let's move on to oxidation. We can't have oxidation without first going through glycolysis. So you have to always remember that glycolysis of one molecule of glucose is 8 ATP. Glycolysis of one molecule of glycogen is 9 ATP. They both end up at this decision tree. We can have either 2 pyruvic acid molecules or 2 lactic acid molecules. If we have these 2 pyruvic acid molecules, they both become acetyls, and from there we can start oxidation. Acetyl enters the mitochondrion. The mitochondrion is where oxidation takes place. We have a number of steps along the way to get into the citric acid cycle, the TCA cycle, the Krebs cycle. There's a lot of names for it. Once we get into this cycle, we end up producing 15 ATP overall. Now remember, back here we had 2 branches. Each branch produces 1 TCA cycle. So overall, we have 2 TCA cycles from one molecule of either glucose or glycogen, and this gives us an additional 30 ATP. That's key. 2 Krebs cycles from one molecule of either glucose or glycogen. So when we put that all together, we have 30 ATP from oxidation with our initial 8 or 9 from glycolysis. That gives us 38 or 39 ATP total at the end of oxidation. Finally, beta oxidation. Beta oxidation is a little different. Here we see epinephrine again. If you remember, epinephrine brought glycogen from its stored form and put it into glycolysis. Epinephrine also acts on fat cells. Primarily what it does is it stimulates lipase to start cleaving free fatty acids off of glycerol, or any fat molecule for that, any fat cell for that matter. The lipase cleaves the free fatty acids, they enter the bloodstream. Remember, we can measure fat in the blood. From there, the fat is brought into the cytoplasm of the cell and then into the mitochondria where it undergoes beta oxidation. Now this is a very complex process. We're not going to get into that. We're not going to get into the electron transport chain or this conversion of NADH, FADH into ATP. All you have to know is that beta oxidation enters the mitochondrion and produces a lot of ATP. But the body likes to avoid breaking down fat. It likes to keep its fat stores. So when we put it all together again, hopefully you get a better picture of how metabolism is integrated, how it's regulated by both hormones and enzymes, and how we end up producing a whole lot of ATP with a little bit of an investment.